Hello guys, my name is Ricardo Catalan and I'm doing my video presentation over cloning. So what is cloning? Cloning is a replica or identical copy of the same DNA of human beings, same skin cells, and usually everything's exactly the same. So one of the things I'm going to talk about is artificial cloning. Artificial cloning is a process of producing genetically identical individuals of an organism either naturally or artificially. In nature, many organisms produce clones through asexual reproduction. The term clone was invented by J.B.S. Scott. Uh, Natural cloning is another one of the terms that I'm going to talk about. And what is natural cloning? It's a sexual reproduction, a new individual generated from a copy of single cell from the parent organism. Natural clones are also known as identical twins. For example, I'm an identical twin. I have my own twin, so you could say I'm a natural clone. Um, it occurs in mammals and in humans as well. Um, the twins are produced um, through a fertilized egg, creating two or more embryos. It could be triplets or even more. And they usually carry almost the same identical DNA, but not exactly the same. Since it's natural, it's not artificial or nothing like that. So usually it's almost identical, the DNA. Another term that I will be talking about is cellular cloning. So it is based on bacteria and yeast naturally produce clones of themselves when they replicate asexually by binary fission. This is known as a cellular cloning. The nuclear DNA duplicates by the process of meiosis, which creates an exact replica of genetic material. Um, another one of the terms that I'll be talking about is vegetative cloning. And basically what that is any form of cloning based on plants. So it is a sexual reproduction of occurring in plants in which a new plant grows from a fragment of the parent plant or a specialized reproductive structure. So anything that has to do with plants that duplicates into other plants. Yeah. So now I'll be talking about the different clones that were made and different clones that were made through animals. And one of them is called Dolly. It is a domestic sheep. It was made in July 5th, 1996, and through February 14, 2003. And it is the first mammal cloned from an adult somatic cell using the process of nuclear transfer. Dolly was part of a series of experiments at the Rosalind Institute that were trying to develop a better method for producing genetically modified livestock. It is successful. If successful, this would mean fewer animals would need to be used in future experiments. Scientists at Rosalind also wanted to learn more about how cells change during developments and whether a specialized cell such as skin or brain cells could be used to make a whole new animal. These experiments were carried out at the Rosalind Institute by a team led by Professor Sir Ian Olmert. Because of the nature of the research, the team was made up of many different people, including scientists, embryologists, surgeons, vets, and farm staff as well. Dolly was cloned from a cell taken from a mammary gland of 6-year-old Flynn Dorset sheep and an egg cell taken from a Scottish blackface sheep. She was born to a Scottish blackface surrogate mother on July 5th, 1996. Dolly's white face was one of the first signs that she was a clone because if she was genetically related to her surrogate mother, she would have had a black face. Because Dolly's DNA came from a primarily a gland cell, she was named after the country singer Dolly Parton. So you may ask, why is Dolly so important? Dolly was important because she was the first mammal to be cloned from an adult cell. Her birth proved that specialized cells could be used to create an exact copy of the animal that they came from. This knowledge changed what scientists thought was possible, and opened a lot of possibilities in biology and in medicine, including the development of personalized stem cells known as iPS cells. However, Dolly was not the first cloned mammal. There's more mammals cloned. Um, the first person cloned animal clone was born in 1984 in Cambridge, UK, which were two other sheep, which were Megan and Morgan. Um, they were also cloned in the lab at the Roslin Institute at Roslin Institute, and other 6 sheep were also cloned there in 1995. Dolly was announced to the world on the 22nd of February 1997 to a frenzy of media attention. The Roslin team chose to make the announcement at this time to coincide with the publication of the scientific paper, which describes the experiments that produced her. Dolly was captured the public imagination, no small feat for a sheep, and sparkled the public debate about the possible benefits and dangers of cloning. When Dolly was 1 year old, analysis of her DNA showed that her telomeres were shorter than would be expected for a normal sheep of the same age. Telomeres are a cape on the ends of DNA molecules that develop the DNA from damage. So Dolly was a very special sheep. She was the first mammal sheep to be cloned, because she was cloned from a adult mammal cell. Every other clone that was made before her were either cloned from small mammal cells, so she was the first. So that's why she was so important, and many people were very eager to see her because of how she was cloned. The next animal that I'm going to talk about is Snuppy the dog. It is an Afghan hound, the first dog clone. The puppy was created using a cell from an ear from an adult Afghan hound involved in 123 surrogate mothers, of which only 2 produce pups. Snuppy being the sole survivor. The Department of Technology at the Sinlo National University, which cloned Snuppy, was led by Woo Suk Han. Snuppy has since been used in the first known successful breeding between cloned Canis, and his sperm was used to artificially inseminate 2 cloned females. So as you can see, Snuppy was a clone dog and was the first clone dog. That's why it's so special for so many people. And yeah. So another person I'm going to talk about is Hwang Woo Suk. He is a South Korean Vietnamese, and a researcher. He was a professor of the same institute as the Snoopy dog, and he was the one that cloned Snoopy the dog. So he was really special in that, and he became very famous, and he had so much pride. He was the pride creator because of that. So he has a lot of respect when it comes to cloning and all that. So so another thing that I'll talk about is somatic cell nuclear transfer. It is a laboratory strategy for creating viable embryo from a body cell and an egg cell. The technique consists of taking an nuclear X cell and implementing it in a donor nucleus. So as you can see, it is an egg cell that is donated to a donor nucleus, and from there it is cloned. So in conclusion, what I think about cloning is that it's pretty cool and can be weird because you don't want to have the same exact replica as anyone else. You want to be your own self. And in reality, I don't think that any human will be cloned anytime soon, but it is possible that maybe in the future it could happen. A human being could be cloned. So yeah, thank you for looking at my research presentation, and yeah, thank you.